Hello there, all future MedTech professionals. Welcome back again to another episode of the Biochem Crash Class, where the complexities of biochemistry are transferred into digestible bites of fascinating facts. The topic of today's episode? Well, it's how the genetic code builds the blueprint for your proteins. Yeah, those ones. The one running your cells, your enzymes, essentially everything that makes up you as a functional human being. By the end of this crash class, you should be able to identify the structures of the DNA and RNA, explain the functionality of nucleotides in your cells, decode codons, and finally, understand how all of that connects to your proteins. So, without further ado, fellow students, let us delve into it! Before we delve into the topic of today, let us first recall the previous topic by asking a question: what does DNA and RNA look like, and what are they made of?
DNA and RNA is made up of a nucleotide, which has three parts: a phosphate group, a nitrogenous base, and a sugar. The sugar tells us a lot because DNA and RNA use different ones. But that begs the question, what sugar tells us if it's DNA or RNA? Well, that table is exactly right. And here's the cool part. Those bases you see, they form the language of life. As you can see, deoxy means without oxygen. And that one change now affects structure and stability. Now, let's say you see this mRNA or messenger RNA sequence, AUG, CUU, GCA. What is that? That's a code, specifically a codon code. And it's how your cells know which amino acid comes next in a protein. Let's see if you know what I mean. Can you name each part of the nucleotide? That's correct. A is the phosphate group, B is the sugar, which is deoxyribose if it's DNA, and C is the nitrogenous base. Nucleotides are essentially the building blocks of nucleic acids. And yes, its structure matters.
This mRNA strand, well, it's not just a random string of letters. It's a code, a codon to be exact. It essentially is a set of three bases that tells your body which amino acid to add. Now, what does that represent exactly? Yes?
Teacher, it's an mRNA strand with codons!
Exactly, random student. Yes, mRNA acts as a messenger from DNA to protein. Now, let's see how that message becomes a protein.
If you remember, DNA and RNA are long chains of nucleotides made up of a sugar, a phosphate, and nitrogenous base, with the sugar in DNA being deoxyribose and in RNA being ribose. Those bases are different as well. DNA uses A, T, G, C. RNA uses A, U, G, C. Notice that RNA swaps out thymine, or T, for uracil, U, because it is a lot more stable in the structure of RNA rather than DNA, since RNA is not a permanent storage of genetic information. Now, let's decode what we learned just a while ago. So, the cell reads mRNA 3 letters at a time. Each codon tells the ribosome which amino acid to add. It goes like this. AUG is the start codon, which brings in the amino acid methionine. Stop codons, UAA, UAG, UGA, tell the ribosome that it is time to wrap it up or stop building.
Now, let's see if you're really paying attention to the lecture then, shall we? All of you viewers at home and random student in the audience, it's pop quiz time! Translate this mRNA. Audience? Random student? Where did he go? Are any of you there?
Our answer is methionine, leucine, and alanine.
Where on earth did you come from? What? Well, moving on from that mini heart attack. Yes, that's correct. Good work, audience at home and random student. Where did he go again? I can't take this.
Now, what you see before you is the protein synthesis chain. But how does this chain lead to protein exactly? Well, here's the flow. DNA is transcribed into mRNA through a process called transcription, where an enzyme called RNA polymerase will connect complementary RNA bases to the DNA, which are then bonded to form mRNA. Step 2 is when these mRNA codons are translated into amino acids through a process called translation. where the mRNA directs the addition of amino acids to the ribosome via the tRNA, or the transfer RNA. These amino acids then form a polypeptide chain as the last step, which is the primary structure of the protein. Now, knowing that, what happens if one base changes or is removed in the DNA sequence? Well, interestingly, you could actually get a different amino acid. This process is called a mutation, specifically a point mutation, sometimes harmless, sometimes completely life-altering, including changes in the gene expression or alterations in encoded proteins.
Actually, fun fact, the 37 trillion cells in our body will acquire trillions of point mutations. These changes come from random errors in copying DNA because of the cell either dividing or from environmental exposures like sunshine or smoke. I know it sounds a little scary, but don't worry. All of these changes are in parts of your genome where it does not really matter. Now, before ending our lesson here, it's now time for us to apply it, or else all of my effort here will have been fruitless. So, let's try this then. Write a 9-base DNA sequence, transcribe it into mRNA, then translate it using a codon chart into a protein. I heavily encourage you to pause the video and try it. Not only does this check your understanding, but helps turn theory that we learned into practice. And with that out of the way, that's it for today's episode of Biochem Crash Class. You've cracked the code, literally the code of your genetics and the proteins that make up your being. Now, if this helped you make sense of the complex concepts of codons and proteins in any way, then make sure you hit that like button and share with your fellow medtech students. They're probably suffering as much as you in biochem. And don't forget to subscribe for more. By the way, this video does not do enough justice about the other concepts of biochemistry, so please also check out our other videos on the channel hosted by our fellow colleagues. Well, this has been Professor J. I hope to see you all next time.